Hello, my name is Mukrama Afoda,
My name is Bushra Ali.
And my name is Muntas Osman.
and our presentation is on Paws, Hooves, and a Whole Lot of Poop, a multi-animal parasite study. This study focuses on zoonotic infections and their transmission between pets, livestock, and humans. Zoonoses can have serious implications, especially for vulnerable populations, such as the immunocompromised, pregnant individuals, and the elderly. As pet ownership is common among students, with 84% of college students having pets, our aim was to determine the prevalence of parasites in various animals associated with University of Minnesota Rochester, students, and their communities. Our primary aim was to elucidate the percentage of UMR students, pets, and in livestock infected with parasites. To achieve this, we collaborated with Mayo Clinic parasitologists to ensure accuracy and adherence to standard laboratory protocols. Zoonotic infections involve bacteria, fungi, viruses, or parasites transmitted from animals to humans. These can occur through direct contact, such as licking or biting, or indirect contact with contaminated surfaces. Alarmingly, in the US, it's estimated that 34% of dogs and a significant proportion of cats may carry parasitic infections. While vaccinations and hygiene practices mitigate transmission risks, it's crucial to understand the parasitic load in our immediate environment. This understanding can help reduce the risks of zoonotic diseases. Our research provides insight into how prevalent these infections might be within our community.
To investigate parasitic infections, we followed a four-step methodology. First, we collected at least 2 grams of stool sample from various animal species, including cats, dogs, sheep, cows, chicken, and horses. Next, we mixed the stool with EcoFix preservative and homologized it thoroughly. The mixture was then processed in the centrifuge at 2000 RPM for 10 minutes to separate and concentrate the parasites. Finally, a drop of the concentrate was placed on a microscope slide for examination. Using 10x magnification, we were able to identify hemolymph eggs and larvae. And with 40x magnification, protozoa were examined. The methodology allowed us to visualize and identify parasites such as mites and giardia, as well as toxoplasmosis in cat samples, and hookworm and entamoeba in sheep samples. By adhering to the standard protocol, we ensure that the reliability of our findings.
Now on to the results. In our study, we examined stool sample from 11 cats, 1 chicken, 2 sheep, 1 horse, and 1 cow. Parasites were identified across multiple species. For example, Giardia and toxoplasmosis were prominent in cats, while sheep samples revealed hookworm and entamoeba infections. Interestingly, 2 out of the 12 cats and 1 out of the 2 sheep had parasites. This node link diagram visualizes our findings, illustrating the distribution of parasites across the whole species. The study also highlights the zootonic risks posed by common household and livestock animals. In conclusion, our findings emphasize the importance of routine veterinary care and preventative measures, such as vaccinations and proper hygiene, to minimize zootonic risks. The study also underlines that the need for increased awareness of parasitic infections, especially in communities with high prevalence of pet ownership.
We'd like to thank Mayo Clinic parasitologists for their invaluable assistance and guidance throughout this project.
Additionally, we are grateful to our team at UMR for their support and participation in this research.
Thank you all for your attention and would be happy to answer any questions you may have.